Have you ever felt like you're just uh, drowning in new scientific information? Especially in fields like genomics, moving so fast.
Yeah.
Keeping up feels almost impossible sometimes, right? Trying to figure out what actually
Oh,
matters.
absolutely. It's a
Yeah.
huge challenge. I mean, the pace of discovery in biology is just relentless. Our tools, they constantly need to evolve to keep up with well, the incredibly rich data we're generating.
Exactly. So today we're taking a deep dive into something that really shook things up, single-cell RNA sequencing, scRNA-seq. It gives us this amazing look at individual cells, but, and here's the kicker, even with all that data, we've apparently been missing something, a whole layer of information just overlooked.
And that overlook layer, yeah, it's exactly where we're going today. We're going to unpack a really groundbreaking method called DOLPHIN. It stands for Deep Exon-Level Graph Neural Network for Single-Cell Representation Learning and Alternative Splicing. Bit of a mouthful, I know.
Definitely.
But the core idea is that it advances single-cell analysis beyond just looking at whole genes. It leverages much more detailed information, exon reads and junction reads.
Okay, so think of it like this, maybe. If our current view of a cell is, say, a clear picture, Dolphin is like upgrading to an ultra-high-res lens.
That's a great analogy, yeah.
So our mission today is to understand why this higher resolution matters so much, how Dolphin actually pulls it off, and what incredible new things it's showing us about, disease and how cells work. Let's get into it.
Okay, before we jump straight into Dolphin, maybe let's just quickly refresh. Why was scRNA-seq itself such a huge deal in the first place? What does it actually do?
Right, good place to start. What's truly transformative about cRNA-seq is its ability to profile gene expression at the level of single individual cells. Historically, right, we analyzed tissue samples. That meant getting an average snapshot from millions of cells all mushed together.
Ah, the average view. Exactly. Now, we can see the unique molecular state of each cell, and that gives us this unprecedented resolution into the sheer diversity within tissues, even tissues that look uniform.
So instead of just the average, you're getting the individual story of every single cell. That must have opened up so many doors.
Oh, absolutely. It's driven huge progress.
Yeah.
Think developmental biology, how a single cell builds a whole organism. Immunology, seeing how different immune cells react. And of course, cancer research, identifying tiny populations of cells linked to disease that we just couldn't see before. It fundamentally reshapes biology and potentially medicine, too.
Okay, so that's the revolution. But you mentioned a blind spot. What were we missing, even with this amazing tech?
That's the critical point, yeah.
Yeah.
Despite all these advances, most standard cRNA-seq analysis is still, well, predominantly gene level. What that means is researchers basically aggregate all the data for an entire gene into just one number, single count.
Okay.
And while that's useful,
Yeah.
it really oversimplifies the picture. It masks crucial information because it treats a whole gene like one single thing.
Yeah, right. It's like trying to understand, I don't know, a complex machine by only knowing if it's on or off, instead of seeing how all the little cogs and wires inside are working together.
That's a good way to put it. We're missing the details, like how specific parts within a gene the exons are expressed, and crucially, how they're actually connected or spliced together.
Okay, so the hidden data, that's these exon-level reads you mentioned,
Yeah.
and junction reads.
Exactly. Exon-level reads map within specific gene segments. And the junction reads, these are fascinating. They actually span the boundaries between exons. They directly capture how these pieces are connected. They're like the blueprints showing the exact connections.
And this connects to another big challenge, right? Alternative splicing. What is that, and why was it so hard to see with the old methods?
Right. Alternative splicing. It's a fundamental process. Basically, different combinations of exons from a single gene can be stitched together to create different protein versions, or isoforms. Think of it like Legos. One gene provides the set of bricks, but you can build different things, a house, a car, by using different combinations of those bricks and connecting them differently.
Ah, okay. One gene, multiple possible proteins.
Precisely. And it's crucial for cell function, for regulation. But the problem is that gene-level counting just obscures all that variation. You can't see the specific isoforms or which exons are being used. Existing tools try to use those junction reads, but they're often really sparse, especially in the common droplet-based methods like 10x genomics. And critically, they often just ignored the much more abundant reads within the exons themselves.
So a ton of really vital information about how our cells are actually functioning was essentially hidden because the tools just weren't sharp enough to see it properly.
That's the situation, yeah.
Okay, before we move to the solution, let's just quickly recap this problem.
Sure. So to sum it up, Conventional single-cell RNA-seq analysis, it mostly relies on gene-level counts, aggregating everything. This means we've been missing really crucial fine-grained details hidden in the individual exon reads and, importantly, those junction reads that show connections. And that limitation directly impacts how well we understand cell states, makes it incredibly difficult to actually detect critical alternative splicing events.
Which brings us neatly to DOLPHIN. It's pitched as this powerful deep learning approach designed specifically to solve this, right? By integrating both exon and junction reads for a much higher res view.
Exactly.
And the promise is better cell representation, more accurate grouping of cells, and finding these subtle biological differences we were missing before. Okay, let's dive into DOLPHIN itself. Sounds like a pretty big leap. What's the core innovation?
The core innovation, really, is moving beyond that gene-level blind spot. It's a deep learning method, as you said, that explicitly integrates exon-level and junction read data. So instead of just summing up activity for a whole gene, DOLPHIN actually, it represents genes as graph structures at single-cell resolution.
Represents genes as graphs. Okay, that's a powerful concept. Like, if a gene is a complex blueprint, DOLPHIN is mapping its internal architecture within each cell. How does it actually do that? What do these graphs look like?
Yeah, the key idea is representing each gene as a graph. Here's how it works. The individual exons, those coding segments within the gene, they become the nodes in this graph.
Okay, the building blocks.
Right. And the splice junctions, those reads spanning the boundaries, they become the edges connecting these nodes. Think of them as the pathways or connections between the blocks. And importantly, these edges are directional, showing the flow of splicing, and they're weighted by the number of junction reads supporting that connection. So stronger connections have thicker lines, essentially.
Wow. So for every gene in every single cell, DOLPHIN is drawing this unique, detailed architectural map, and then it feeds these maps into some kind of AI.
Precisely. It uses what's called a variational graph autoencoder, or VGAE. It's a type of deep learning model specifically designed to learn rich information from data structured as graphs. This allows DOLPHIN to generate much more accurate and informative cell embeddings. These are basically numerical fingerprints that capture the unique characteristics of each cell based on this detailed graph structure.
And the initial results.
The early impact looks really remarkable. DOLPHIN shows superior performance in cell clustering, grouping similar cells much more accurately. It's also proving excellent for finding biomarkers and, crucially, for detecting that alternative splicing we talked about.
This ability to find, quote, subtle transcriptomic differences at the exon level that were previously masked, that has huge implications, doesn't it?
It really does. It means we get new insights into disease mechanisms and potential therapeutic targets. By seeing these subtle, previously hidden changes, we can understand disease progression at a much finer level. That could lead to new diagnostic markers, maybe new treatment strategies.
That's really compelling. Okay, let's break down how DOLPHIN actually does this. The paper outlines 3 main steps. Can you walk us through them?
Absolutely. It operates in 3 main steps, each building on the last, to boost that transcriptomic resolution. Step 1 is constructing gene-specific exon graphs. This is where we build that detailed architectural blueprint for each gene. It starts with pre-processing. You take the raw scRNA-seq, reads those short genetic sequences, and align them to a reference genome. During alignment, DOLPHIN flags 2 key types. The exon reads that map entirely within an exon.
Okay.
And the junction reads that span the boundaries between exons. These junction reads are direct evidence for how exons are actually connected or spliced.
Got it. So not just counting reads per gene, but pinpointing where within the gene they fall or if they bridge 2 parts.
Exactly right. Once those reads are identified, DOLPHIN builds an exon graph for each gene in each individual cell. In this graph, like we said, each exon is a node. Think of it like rooms in our architectural plan. And each node is annotated with its read count, so how much activity is happening in that specific room.
And the connections between the rooms, the corridors.
Those are the edges representing the connections shown by the junction reads. They're directional, showing the flow, like A connects to B, and they're weighted by the normalized count of junction reads. So a heavily used splicing path gets a stronger edge weight. This whole process creates this incredibly detailed cell-level structure of exon graphs for each gene.
Okay, so we've got this super detailed blueprint for every gene in every cell. What next? How does that help us understand the cell better?
Well, that leads directly into step 2. Learning informative cell embeddings with a variational graph autoencoder. This is where DOLPHIN creates that unique digital fingerprint for each cell. These detailed exon graphs, represented mathematically, get fed into this VGAE framework. It's a powerful neural network designed specifically for learning from graph data.
And there's a key component in there, right? A graph attention layer, or GAE. What's its role?
Yes, the gate layer is crucial. It dynamically assigns weights to neighboring exons. Think of it like a smart filter on our gene blueprints. It doesn't just see connections. It learns to emphasize the biologically relevant ones informed by those junction reads. It figures out which exon-to-exon connections are most important for defining a cell's specific state.
Okay, so it's learning the important pathways.
Exactly. The VAE's encoder part then takes all this and learns a latent representation Z. This is a complex, multidimensional vector, a compressed, highly informative summary that captures those critical exon-junction relationships.
And then there's a decoder part, too, to make sure it learned the right things.
Correct. Two decoders, actually. They work to reconstruct both the original exon-level features and the graph structure, the adjacency matrix. This forces the model to preserve both the structural and attribute information. So this final latent representation Z, this vector, it's like creating a highly detailed, multidimensional digital fingerprint for each cell, based purely on its unique gene architecture and splicing patterns. No two are exactly alike.
Wow. So with such a precise cellular fingerprint, what new discoveries does that unlock? What could we see now that was hidden before?
Well, this takes us neatly to step 3, aggregating junction reads for enhanced alternative splicing detection. This addresses that persistent challenge we mentioned earlier.
Sure.
Single-cell data often has really sparse coverage of junction reads. It's hard to see every connection in every cell.
Right, the data can be patchy. So how does DOLPHIN fix that? It can't just invent data, can it?
No, it doesn't invent data, but it does something clever. It aggregates junction reads from similar cells. It identifies using that learned junction-aware latent space from step 2. It uses a k-nearest neighbors approach, KNN. It finds, by default, the 10 cells, K10, that have the most similar exon splicing patterns based on those learned fingerprints.
Okay, so if cell A's splicing info is a bit incomplete, DOLPHIN looks at the 10 cells most like cell A and pools their information.
Precisely. It then performs majority voting. If a specific splice junction appears in more than half of those neighbors, plus the original cell, DOLPHIN confidently adds that junction information to the original cell's profile. This aggregation step enriches each cell's profile with junction reads, dramatically boosting the sensitivity for detecting alternative splicing events, crucially, without just adding noise. It's like confirming a faint signal by seeing it pop up consistently across similar neighbors.
That makes sense, like crowdsourcing the details from similar cells, and the result of this enhanced data.
The result is that we can then calculate PSI values percent spliced in much more reliably using standard tools like Outrigger. This gives us accurate and robust quantification of splicing events. We can finally get a clear, quantifiable measure of which splice variants are active in which specific cells. That was largely guesswork before.
Okay, that's a lot to take in. Let's quickly summarize that 3-step process.
Right. Step 1, the blueprint. DOLPHIN builds those detailed exon graphs for each gene in each cell. Exons are nodes, junctions are weighted edges. Captures the architecture.
Step 2, the fingerprint. A variational graph autoencoder with that key graph attention network learns a unique multi-dimensional latent representation or embedding for each cell, capturing complex exon-junction relationships.
In step 3, community wisdom. To beat data sparsity, DOLPHIN finds similar cells using KNN and aggregates their junction reads via majority voting. This massively improves alternative splicing detection.
Okay, DOLPHIN sounds fantastic in theory, powerful stuff. But what about the real world? The proof is in the pudding, right? How does it actually perform compared to the older methods?
Ah, yes, the results. This is where it gets really exciting. They validated DOLPHIN on several different real-world scRNA-seq datasets. Things like full-length human peripheral blood mononuclear cells, PBMCs, and also common 10x genomics data from human colon and rectum tissues, including from cancer patients. And the results consistently show superior performance.
What's the most obvious difference when you look at the data?
Visually, one of the most striking things is in enhanced cell embeddings and clustering. If you use those UMP plots, the 2D maps we use to visualize cell populations, DOLPHIN consistently gives you clearer, more distinctly delineated cell clusters, much sharper than traditional gene count methods.
Right, so instead of fuzzy blobs where cell types kind of merge, you see really distinct groups. Like in the PBMC example, it could separate out T-cell subsets that were all mushed together when just using gene counts.
Exactly that. And it's not just visual. Quantitatively, DOLPHIN scores significantly higher, on metrics like ARI, adjusted RAND index, and NMI, normalized mutual information. These numbers measure how accurately cells are grouped. For PBMCs, the median ARI was 0.11 higher. That's a statistically significant jump, meaning much more reliable cell grouping.
What about batch effects, those annoying variations between experiments that can really mess things up?
A good question. They can be a real headache. But DOLPHIN's exon-level modeling seems to inherently mitigate batch-driven separation. It focuses on these finer details that are less affected, and it works even better when you combine it with standard batch correction tools, applying them to DOLPHIN's exon-level inputs.
Okay, so if we can cluster cells much more accurately, resolve these subtle subpopulations, how does that change our understanding of tissues and diseases? What does that let us do?
Well, it means we can identify cell types and even subtle cell states with unprecedented precision. Think about understanding exactly which tiny subpopulation of immune cells is responding to a treatment, or which specific cell state is driving tumor growth. It could revolutionize how we study complex tissues, disease progression, leading to much more targeted interventions down the line.
That makes sense. And beyond just clustering, DOLPHIN also finds these exon-level differentially expressed genes, EDs. Tell us about that.
Yes, this is a really impactful finding. They did a case study on pancreatic ductal adenocarcinoma, PDAC, a really tough cancer. Using DOLPHIN, they uniquely identified 896 EDs changes happening at the exon level that were completely undetected when analyzed solely at the gene level. These are critical changes that were essentially invisible before.
Whoa, 896 hidden gene changes. That's like finding microscopic cracks in a foundation, like you said, that a regular camera misses, but they're crucial for predicting failure. What kind of genes were these? Anything relevant?
Highly relevant. These unique EDGs showed significant enrichment of pancreatic cancer-related terms in pathway analyses. Things like SMAD4, a well-known tumor suppressor often mutated in PDA, ERCC1, linked to chemo response, TGFBR2, involved in tumor signaling, ATM, a DNA damage response kinase. These crucial players showed exon-level changes missed by standard methods.
And finding these previously hidden markers, does it actually mean anything clinically?
It seems so. They performed Kaplan-Meier survival analysis using data from the Cancer Genome Atlas, TCGA, for PDS patients. And these DOLPHIN unique EDAs showed strong clinical relevance in stratifying patient risk. Using all 896 EDGs gave a very significant prognostic signal. This suggests these newly visible exon-level changes could potentially be powerful new biomarkers for diagnosis or prognosis in the future.
That ability to find these hidden disease markers, that could really change things for diagnosing and maybe even treating complex diseases, couldn't it?
Absolutely. The precision points towards potentially much more specific diagnostics and maybe even tailored treatments eventually.
Okay, and the third big area was alternative splicing detection.
Right. Effective alternative splicing AS detection. And here again, the improvement is dramatic. DOLPHIN significantly boosts the detection of common ANIS events like exon skipping, ES, and mutually exclusive exons, MXE. In that PBMC dataset, for example, the median count of detected ES events per cell jumped from just 183 to 1215 after DOLPHIN aggregation.
Wow, that's a massive increase from under 200 to over 1,200 per cell. What about accuracy? More isn't always better if it's wrong.
Good point. But accuracy improved too. They found a higher correlation between DOLPHIN single-cell PSI values and pseudo-bulk values, which act as a kind of proxy ground truth. This indicates more accurate AS quantification. And again, human plots based on these refined PSI values showed sharper boundaries between cell types, suggesting that the splicing signals DOLPHIN captures are more biologically meaningful.
Can you give us a specific example, a splicing event that was missed before but DOLPHIN found?
Sure. They use these visuals called sashimi plots. In PBMCs, there's a gene called PTPRC. It has an exon skipping event critical for T-cell function. Standard single-cell analysis couldn't see it, not enough junction reads. But after DOLPHIN aggregation, bang, it was clearly visible. Another example in the 10X colon data involved the CD47 gene, which is implicated in colorectal cancer. Again, a specific splicing event became detectable only after DOLPHIN did its magic.
So it's not just finding more splicing, it's finding the biologically important events that help define what a cell is doing or what's going wrong in disease. That seems critical for understanding cell type-specific functions.
Exactly. It reveals distinct splicing patterns not captured by raw single-cell data. When they looked at the biological processes using Jugo terms associated with these newly found splicing events, they made perfect sense. For B cells in PBMCs, you saw enrichment for things like B cell activation, For enterocytes in the colon, it was metabolic processes, respiration. It confirms the biological relevance.
Okay, this is really impressive stuff. Let's do one last quick recap of these performance results.
Sounds good. So for clustering, DOLPHIN gives significantly more accurate cell grouping, identifies subpopulations better, beats traditional methods, and it's robust to batch effects.
For disease markers, it uncovers those hidden exon-level differentially expressed genes, EAGs, like in the pancreatic cancer example, and these have real clinical prognostic value.
And for alternative splicing, it dramatically boosts both the detection and accuracy of AS events, revealing critical cell type-specific patterns that were basically invisible before.
Okay, this feels like a genuinely significant step forward. So what's the bigger picture here, the broader impact, and maybe what's next for DOLPHIN?
Well, summing up the key advances, DOLPHIN offers this high-resolution exon-level cellular representation. It gets past the limits of just counting genes by integrating exon and junction reads. It seems to have broad applicability working on different types of scRNA-seq data, from older full-length methods to the common 10x genomics platform. And its unique strength is identifying those subtle EDGs and cell-type-specific alternative splicing patterns that were hidden from view before.
That sounds incredibly powerful. Are there any limitations right now? Areas where it still needs to grow?
Yeah, like any new tech, there are definitely areas for development. One current limitation is that it's not yet fully compatible with all existing single-cell analysis frameworks, especially tools built strictly for gene-level counts, like maybe some trajectory inference or cell interaction tools. That integration work needs to happen.
Okay, so plugging it into the entire existing ecosystem takes time.
Right. Also, the benchmarking so far focused mainly on exon skipping and mutually exclusive exons. They're the most common AS types, over 50%, but there are others, like intron retention or alternative splice sites, that could be explored more deeply.
Makes sense. So the potential for future integration and expansion is huge, but it's a process. What's the ultimate goal, the trajectory?
I think the trajectory involves exactly that, expanding compatibility with more downstream tools, broadening the range of splicing events it handles robustly. Ultimately, the goal seems to be creating a really comprehensive, high-resolution framework for a much wider range of single-cell analyses.
You know, this really feels like more than just a technical jump. It's almost like a new way of seeing inside cells. By digging so much deeper into the genetic blueprints, DOLPHIN could genuinely revolutionize basic biology, but also how we find personalized treatments or diagnostic markers.
I agree. The precision it offers could really open up entirely new avenues for research for understanding disease mechanisms at a much finer scale. That's exactly what we need for developing better, more targeted therapies.
It makes you wonder, what else is hidden in plain sight in our data, just waiting for the right tools to reveal it? This deep dive into DOLPHIN really highlights that sometimes the biggest insights come not from new data, but from a new way of looking at the data we already have.
That's a great point. So thinking about all this, what stood out most to you today? And what diseases or areas of biology do you think could benefit most from this kind of high-resolution view? We genuinely love to hear your thoughts.
It's discoveries like this that remind us, isn't it? The quest for knowledge is always about peeling back another layer, going just that little bit deeper, and revealing what was previously unseen.